From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 25th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 61st episode of Matters Microbial. I continue to try to find interesting microbial scientists to share their passion for microbial enthusiasm with all of you. Wish me luck in keeping it going. My Bio 350 Micronauts are learning about the history of microbiology with fascinating people like Angelina Hesse, who developed the idea of using agar in petri dishes, to titans of the field like Lynn Margulis, Carl Woese, and Paul Ehrlich. In laboratory, my Micronauts are starting to explore their water bottle buddies with this sticker to inspire them. The microbes are everywhere, including the water bottle refill station seen here. As the students begin to explore their water bottles, some samples are rich in microbes, seen here, while others are more manageable. Our goal, of course, is to streak out individual isolates for analysis, seen here. They're having fun with that, I can promise you. From time to time, we see odd things on these plates. For example, here is what may appear to be bacteriophage plaques, but are in fact gliding bacteria pushing other microbes out of the way. Remember that every environment is just a surface for colonization, not just water bottles, but even ourselves. To learn more about basic microbiology techniques, I enjoy using E. coli containing plasmids with pigment genes, seen here under both natural light and UV. These were developed by former podcast guest Sebastian Kosiaba, and one of my students called them, and I love this, forbidden Skittles. Can't wait to show you photos. Finally, my Micronauts continued to enjoy working with bioluminescent bacteria. Sometimes they get creative, as with this ghostly Halloween-style image. Other times, they pose with a glowing flask. And from today, here is an example of the kind of lux art that students can make, using creative approaches to teach microbial enthusiasm. Many people think of bacteria as living in liquid cultures and petri dishes, but they exist in natural environments, even entering into periods of dormancy, say from drying out, and then waking up as needed. This idea of growth versus non-growth is relevant to many aspects of microbiology, including, believe it or not, medical microbiology. So, I'm pleased to introduce the very interesting Dr. Paul Carini to discuss the work of his research group on this aspect of environmental microbiology. Welcome to the Quality Quorum for today, Paul.
Hi, Mark. Thank you so much for having me today. I'm pretty excited.
I think we'll have a fun time, and we've followed one another on the various aspects of social media for some time.
Yep.
I think that one of the things that I do in this podcast is I like to put up links, and your I'm going to call it your lab group website has such interesting stuff. You have videos about heavy metal microbiology, which has nothing to do, I think, with heavy metals, but more about an appreciation for the music. And you have like a variety of different, I hesitate to use the word quirky. I would rather use the term Carini viewpoints. Yeah. And I hope that everyone will enjoy that. So can you tell the audience of viewers and listeners a little bit about how you came to the microbial sciences?
Sure. Microbiology kind of wasn't on my radar growing up, literally and figuratively, I think. But I had this innate fascination with digging holes in the earth. And kind of as a young kid, you could find me prancing around my yard with a little shovel digging holes. And I lived next to a tavern. In Wisconsin, there are lots of taverns, and I lived pretty close to one. And over the years, some bones from the chicken wings would end up in the dirt, buried in this lot next to our house. And I would go dig them up, and I'd be like, "Oh my gosh, I found dinosaur bones." So I kind of associated um, digging holes in the dirt with discovery at a young age. And I think after high school, I had a decision to make about what I wanted to do in college. Initially, that was going to be physical therapy, and I had to pick a major, and the
Hmm.
major I picked was microbiology. And I kind of
Hmm.
struggled my first year mightily in college and got back on my feet after a couple years. And really, it's been nothing but microbiology um, ever since then.
Can I break in for just a second and remind listeners and viewers how important it was that you said this? Everyone seems to have this idea that every successful scientist had some fabulous background where everything worked. And it's simply not true. I can't speak for you, Paul, but I suffered from a certain degree of testosterone poisoning growing up. So it was very hard for me to ask for help. I had a lot of trouble with my classes, and it took me until about my sophomore year to get my head on straight. And it sounds like you had something of the same experience.
Absolutely. That first year of college, to dig in a little bit, I got kicked out of two universities, two back-to-back semesters. The first one, I just didn't go to class, and I was one of the students that asked for forgiveness with half a semester to go, and then I didn't fix it. On top of that, went to a second university. Same thing happened. Didn't go to class. One that was closer to home, and took some time off. And my guidance counselor at the time had said something like, "We're not going to admit you back to the university until you can prove you can do quality work, university level work." So I went to a tech school for a year and a half and got my feet back under me, and then re-enrolled at that same university I got kicked out of. Took all the classes I failed over again, and really kind of powered through. And I remember when I graduated, that same guidance counselor saying, "I don't think I've ever seen anyone come in here in as bad a shape as you did and graduate." So, there's this immense power for people to fix mistakes that I think doesn't um, get enough attention. And I think that that's something that a lot of people are afraid of, and should be brought to light that, when you're useful, you can fix errors and fix mistakes and go on to bigger and better things.
That is such an important lesson. I want to thank you for sharing it because it's so common to run into students that will say, and this has nothing to do with you or me, I can never go, say, to medical school because I can't get through organic chemistry. None of that's true. They can do it. They're just not in a place at the time where they're ready to do it.
Yeah.
Because let's face it, we all have to face things a lot tougher than organic chemistry, just in
Yep.
everyday life. And I'm
Yep.
not being negative about OChem at all. I'm simply saying that it's a time and a place issue. I mean, I cringe thinking about how I dealt with college, but you just have to not give up and keep working toward your goals. And I think your story is a useful one for people to hear. So thanks for that.
Yeah. Absolutely.
So the subject for today is going to have something to do with this dormancy issue. You have a wonderful website. I already explained that I'm really impressed with it, and there's going to be a link in the show notes so people can observe it themselves. Um, I thought you might want to. This is up to you to talk a little bit about the work that your research group does, and then
Hmm.
focus in on the project of today.
Yeah. So my passion, and I think the thing that brought me to microbiology, was this fascination with trying to culture or almost even conjure microbes from the environment in the lab. And I don't remember specifically when I got fascinated with that, but I remember linking certain names with that when I was in college and doing my master's degree. And that passion really drives most of what we do in our lab, whether it's directly related to the research that we're studying, i.e., maybe we have a project where we're trying to culture things, or if it's there's a direct line to it. And when we think about dormancy and we think about different forms of dormancy, um, that's directly related to our ability to culture things, where we have to wake them up from their dormant state in order to get them to grow in the lab. So the general framework that our lab group uses when we investigate any problem in any environment is something that we um, call microbial systems ecology. So if you kind of take a step back and think about microbiology as a discipline, there are almost two kind of camps. There's the side of things that focuses on top-down approaches to understand the role of microbes in the lab, in the environment. Um, and these kind of tools nowadays are dominated by microbiomes and metagenomics or other metaomic techniques, but they also include bulk rate measurements, enzyme activities, things like isotope um, studies and biomarker studies. And all of these approaches kind of have this goal of understanding the role of microbes with a capital M in the environment. And often they're a little black boxy. A lot of the measurements you measure at the beginning of a treatment and at the end of the treatment. And the difference between those two is the result of microbial activity.
Hmm.
Microbiomes and metaomics are a little bit um, they're not as black boxy because we can see lots of things, but they are just snapshots in time of whatever the community composition is or whatever the um, metagenome composition is. So these tools, as many of the people watching this probably know, have completely revolutionized microbiology, especially in the last 15 years or so. But there are some pretty clear limits to what they can tell us about an environment. They can't always tell us who's doing what. Um, they are very often at inappropriate scales. So the microbial scale
Mm.
happens at micrometers. So when you extract DNA from a gram of soil, there are lots of micrometers in a gram of soil. So we're really getting this almost artificially large picture, and we don't always know what to measure. So we measure the things we can measure at the scales that we can measure them, and those may or may not be appropriate markers for microbes at the microbial scale.
Can I break in for a quick comment?
Yeah.
I love this a lot because scale is so important. And I remember a million years ago when I took the Woods Hole course in microbial diversity, we had an oxygen sensor with like this very fine needle, and we had wonderful controls for it. And we're measuring plenty of oxygen until we touch the surface of a colony on a plate and it drops to almost nothing, which tells you that inside the colony is a very different environment than right outside the colony on a very fine scale. In a similar way, and I hope that if I'm putting this in artfully, you'll forgive me. A lot of people who study the human microbiome or animal microbiomes look at fecal samples as a snapshot of what's taking place in the gut. And I'm not sure, I don't want to be dogmatic. I'm not sure that really answers the question quite the same way. It does tell you what's taking place in the fecal matter, but in the epithelia, I'm not so sure. But it's what we can get more easily. So this like drives home the point that you just made, is that we tend to not appreciate the fine level at which things are taking place.
Yeah. The fine level, we do things that are relatively easy, I think. And there's a span of difficulty along the word relatively in that context. Um, yeah, so that's kind of the microbial ecology perspective of things. So we need other approaches to really get at microbial function, how genes are expressed in certain conditions. And that's when sort of these bottom up approaches, um, what we would consider maybe classic microbiology, come into play. Things like culturing cells in the lab and studying their physiology and studying how they're using their genomes using functional genomics tools. And there's a whole list of different functional genomics approaches and tools that are now available. These approaches are very good at what I call pinning the tail on the donkey, right? This gene is involved in this process under these conditions. And it's really been really helped us figure out how cells work over the last 50 years, because this is maybe a little bit of an older discipline.
Hmm.
So, what our lab tries to do, I should note that that's also not perfect, by the way. Systems biology is not perfect. The lab is not the environment. It's this nice, cozy environment for the microbes where food is plentiful, predators are non existent for the most part. There's very little, if any, competition depending on the scenario. And we know that we can't culture everything, or most things have not yet been cultured, is like how I like to think about it. Um, so this is not a perfect solution to the problem. So what our lab tries to do is bounce back and forth between these top down and bottom up approaches to try and span this continuum of scale from the molecular scale of DNA and individual metabolites to the ecosystem scale of, a land a land slope or something like that, landscape, sorry, or something like that. So we go back and forth. Sometimes we start on the top down side. Sometimes we start on the bottom up side.
And you're located in Tucson.
Yes.
And I believe you have a wonderful sampling photograph of when people are out taking samples. I don't know if it's relevant to the discussion today, but it certainly gives you kind of a feel for it, because when I think of Tucson, that's not what I think of. So I'm delighted you put that in there.
Yeah. Yeah. Sampling here, depending on when you go, can be pretty difficult. And that particular picture is taken in, I believe, August, which is not our hottest time of the year, believe it or not, but it is very hot and pretty humid. So I think it was probably 100 degrees Fahrenheit and probably 70% relative humidity or 60%.
Amazing.
That's in the middle of our monsoon season. So when I first arrived in Tucson and you look around, everything looks dead. I arrived in June, and that's our hottest time of the year. Very hot, very dry. And as I found myself pondering this foreign environment from inside my air conditioned house, I started to wonder, like, what do the microbes do when it's this hot and this dry out? How do they survive and persist in the desert? When the rains come in July, everything wakes up. It's kind of crazy. Within 2 or 3 days of the first rain that we get in July, all of the trees leave out. All of the cacti start to get plumper and a more vibrant, almost neon green color. And really, everything in the desert that we can see above ground is designed to hang on to their water. So I started thinking, like, well, maybe the microbes here have some tricks up their sleeves to cope with this really hot, dry season.
And what did you find?
To start asking these questions, we needed or wanted to get a collection of microbes. So we spent the first year and a half of our lab group constructing this culture collection. We call it the Arizona Culture Collection. We went all over the local environment at sites that were part of the Critical Zone Observatory that was formerly funded by NSF. These are sites that have been studied for a few years, and they're highly instrumented for various um, biological and geological attributes. We kind of piggybacked on that because there were some nice data sets available and used a variety of techniques to culture microbes from these environments. We used dilution to extinction for the first time. That was a paper published by our postdoc at the time, Ryan Bartlemy. Um, and then we used a variety of other more traditional plate-based high throughput culturing methods. We now have something like 3,000 cultures from these handful of sites, including some archaea as well now. And um, it's a very diverse culture collection. So on this slide here, we have a picture of the phylogenetic reconstruction of the lineages that are in this culture collection. So there are lots of different taxa. And there are also, in some of those lineages, lots of redundancy within each phylotype. So, for example, Bradyrhizobium, we have something like 100 cultures of Bradyrhizobium from these different soil sites. So it's a really taxonomically diverse, but also redundant data set that allows us to ask questions like, how do different taxa cope with desiccation? How different taxa from the same environment cope with desiccation? And how do related microbes cope with desiccation? And this question has really not garnered that much attention yet, because the assumption is that all microbes do respond to this stress the same way. So we're very interested in whether or not there are different mechanisms of desiccation tolerance within specific lineages of microbe specific phylotypes.
It's interesting to hear you say this because we tend to, when we think about microbes, mostly we have people before they think about microbes that just, ooh, germs, that kind of stuff, which is normal. You can't really blame people for that. And then when you start to work in a lab, you start to think of them growing in culture tubes or on petri dishes. That that has nothing to do with reality of life where they normally live. We also put our focus on things that are quickly growing or very happy, and that also doesn't probably reflect what you normally see. So, this idea of how microbes deal with stress is important. And there are different types of stress, as you see. And I want to emphasize for listeners and viewers, you may think, well, those are desert microbes. But there is a broad amount of information out there suggesting that certain types of bacteria that cause disease can enter into dormant states. So I can't help but think that what you're learning here about desiccation tolerance may shed some light on things that might even be of medical importance.
Yeah, absolutely. Desiccation tolerance is one of these stresses, I think, that doesn't always get the attention across a lot of disciplines that it maybe deserves. So, yes, soils dry out in the desert. They also dry out in other regions of the country. A drought is a lack of water, and there are different regions of the country go through this periodically. Um, Airborne microbes, depending on their environment, are dried out. So blowing dust in Tucson, yes, is very dry. Blowing dust in Atlanta is maybe not as dry. There's a little bit more humidity there. Shout out to all you Atlanta folks. And maybe the responses are different. There are also astrobiology implications to desiccation tolerance. How do microbes survive on other planets with no water or little water or frozen water? Um, and a lot of diseases, plant diseases, are transmitted in the air as well and may have relevance, or allergens in houses. I mean, there's all sorts of intertwined environmental impacts of desiccation tolerance.
So, just two things really quickly. When I was teaching in my micro class, we were talking about the sizes of microbes, upper and lower limits. And it so happens that when you starve some marine bacteria, they shrink way down, and they give them the very creative name of ultramicrobacteria. But the interesting point is, it's almost imagine all what I call the central dogma machinery shrink wrapped. So they're not
Yep.
necessarily very metabolically active, but they're ready to go. And it's not making a spore. That's one reason I'm breaking in here, is to say that we're used to the idea of spores. And I find that some students don't know what they are. And spores are a resting stage of some type of, well, you can find them in lots of things besides bacteria. But the point is, they help you go through these kinds of sessions, unusual situations. So this idea of dealing with different stressors, and especially, I mean, I was just thinking about you when I looked at a too old of a plate in my lab and all the dried out colonies. And if you think about it, that's desiccation pressure too, isn't it?
Yep. Yep. We have some of those as well. Um, yeah, desiccation is really fascinating. So I'm glad you brought up spores, because I like, a lot of people, when I talk about this, they say, "Oh, well, the microbes in the soil here." Let me back up just a second. We get rain in the winter here. Um, so we have this really interesting weather system where we get rain from the west in the winter, the same system that brings rain to Southern California in the winter months. And we have a dry period from like March until July. There's no precipitation, very little, very low relative humidities in the teens typically. And then we get a summer monsoon system that brings moisture to the Chihuahuan Desert east of us in New Mexico. And we get a little bit of that. We get the eastern tail of that precipitation pattern. and then it dries out again before we get our um, winter precipitation again. If you were to measure microbial activity in the soil from March to July, it would gradually decrease as it dries out. And then when those rains come, you see a spike in activity and it's relatively high until again it dries out sufficiently that microbial activity um, can't persist. So a lot of folks think, oh, well, the microbes must just form spores. And when you look at the microbiome of these environments, yes, you see spore formers, mostly
Mhm.
actinobacteria and bacilli, but they're
Mhm.
not dominant in terms of the relative abundance in the communities. They're there, they can form spores, but they're not the only microbes that are present. There are definitely non spore formers present. Sporulation is this cell differentiation process that has very specific cues that when cells are starved, typically, they go into this resting state. And yes, they're desiccation tolerant. But to my knowledge, there's no example of desiccation inducing sporulation unless it's also happening at the same time as starvation. And it's also something that's relatively rare in these microbiomes in these environments. So we got really interested in how do non-spore forming microbes cope with desiccation? They're not getting these cues to form this complex cell architecture. They're simply drying out, which is unpredictable in many cases. Um, so we developed some of the microbes that we have in culture into model systems to look at this. We isolated the microbes from one of our field sites here in Tucson, and we honed in on an Arthropactor strain to really try and understand how some of these microbes cope with desiccation. Um, why did we choose Arthropactor? I get this question a lot, and it's nice to grow in the lab. A lot of our strains are difficult. We know it's a gram positive. It's not a spore former. We know that there's literature showing that it can be desiccation tolerant. So it seems like a good system to investigate um, how non-spore forming microbes cope with desiccation. So we grew this up in batch culture in the lab, nothing fancy, filtered it onto filters, and then developed these desiccation chambers to study the biology of slow dried Arthropactor. And the desiccation chambers are just plastic totes. Again, this is work that was developed by our postdoc at the time, Ryan Bartlemy. Inside of those chambers, there's a little beaker that we put either water in or a saturated salt solution in.
Mhm.
And when we put water in it, the relative humidity of that chamber is 100% relative humidity. And if we put a salt in, depending on which salt it is, we can sort of suck the humidity out of the water out of the space. So different salts have different physical properties that allow them to absorb or release water vapor from them. And then we monitor this with a relative humidity sensor in the chamber. So over the course of 2 weeks, we slowly dried these filters that we put in these chambers out. And I believe we have a slide of this, and the red line shows the different salt solutions kind of stair stepping their
Mhm.
way down from 100% relative humidity all the way down to 25% relative humidity over the course of 2 weeks. Now, why did we choose 2 weeks? Um, because most soils take a long time to dry out. The desiccation studies that are out there now take filters, put them in a desiccator, suck all the air out, and dry them out almost instantly within a few hours. We said that's not really realistic for soil. We wanted to do this
Mhm.
slow, controlled dehydration. And the other thing that a lot of studies in the past haven't done is they haven't looked at rehydration. So, what happens when water is reintroduced to these cells? How do they wake up? And this is where the link to culturing comes in. We think that maybe adding soil to a buffer might be really stressful when we initiate culturing experiments. So, we wanted to ask is there maybe a more gentle way of waking these cells up? So after 14 days of these filters sitting in this desiccation chamber, we reintroduced water into that beaker and rehumidified the air. So we're not adding liquid water. to the cells themselves. We're just rehumidifying that atmosphere. And we let the cells sit there for 48 hours. Um, at 4 different time points, we collected some of those filters and looked at how many cells were culturable on the filters, from the filters. Um, what do the transcriptomes look like in the cells at those different states? And what cellular metabolites are present inside of the cells at those states. So we're trying to get as holistic of a picture of the
Hmm.
biology of this desiccated state as we possibly can.
This is very interesting to me on so many levels. I'm an old science fiction nerd. And when the Viking lander landed on Mars, one of the experiments was to take some soil and dump it in nutrient broth to see what happened. And I was thinking about what you just said there about the stressful nature of that sudden change. And the people who designed the experiment were very smart people, but they were thinking kind of like people and not like microbes. And yes, I know microbes don't think, but you know the idea of what I'm trying to say. You want the ecophysiology to kind of reflect what's going on. So I think this is exactly right. And the idea of using transcriptomics to find out what genes are being active during the rehydration, this is something that I really want to emphasize with listeners and viewers. For relatively modest amounts of money, if you have a known genome, you can actually find out what genes are being turned on and off. I'm not going to say easily, but much more easily in the bad than in the bad old days.
Yes, exactly. I'm old school microarray person, so um, I remember some of the days where it was difficult to do these kinds of experiments. More difficult, maybe not difficult per se. Um, so the first thing that we found that it was really interesting is that water vapor seems to resuscitate dry cells. So this was a surprise, I think, for everybody when we started to collect this data. So we take these filters that have been in the chamber for different amounts of time, and they're at different relative humidities, and they're in the chamber that is set at different relative humidities. And we dunk them in a buffer and shake all the cells off. And then we count the cells with a flow cytometer, and then we plate some cells on Petri plates and calculate the fraction of cells in both a hydrated control and a treatment that has been desiccated that are able to be cultured. And what we find is that after 2 days in the chamber at a relative humidity of 100%, so both the treatment and the control are at 100%, that there's effectively no difference in the culturability between that treatment and control, which makes sense. The treatment and control chambers at this point are at identical humidities. But as soon as we start drying out the treatment chamber relative to that control, we see that culturability drop down to 10% or less. So at 65% relative humidity, which is very dry for a microbe, I want to just make that clear. It's very dry for a soil as well. Um, about 10% or fewer of the cells are culturable relative to the hydrated control. And we see the same thing at 26%. So if we keep drying those filters out, again, we see this very low culturability. Now, if we think about what's going on with this, we're taking a filter and we're essentially instantly rehydrating it by dunking it in this buffer. Okay. We think that that's very stressful for the cells. And there's been some speculation in the soil microbiology community, maybe not speculation. There are probably some good studies that show this um, that rehydration may lyse cells because it's a very abrupt physiological stress, and there's a rapid change in
Hmm.
turgor pressure that may lead to the cells bursting.
Hmm.
But if we take those dry filters and rehumidify them with water vapor, so all we're doing is changing the atmospheric relative humidity. That is enough to prime those cells to be resuscitated at levels that are identical to the hydrated control. So none of these cells have access to nutrients. The only difference between the treatment and control is the availability of water on at day 8 and day 14. Um, we were surprised by this. To my knowledge, there's no um, very little, if any, literature describing the use of water vapor by a bacteria to activate their cellular capability to be cultured, whatever that consists of. We started to see some clues where we're like, well, how are they doing this? This was kind of weird. So we started looking at some of the metabolomic data. This was some data collected by a former master's student, um, Izzy Viney. Uh, and what she showed is that there are these compounds in the desiccated cells, and I have two here on this slide, pyroglutamic acid and mandelic acid, that may be involved in this. So, for those of you that use skincare products, mandelic acid is often an ingredient, and pyroglutamic acid is less often an ingredient, but can be found in some of them because it's pretty expensive to manufacture. At least that's my understanding. But they have humectant properties where they're able to absorb water vapor from the environment and bring it to your skin in the case of skincare products to prevent your skin from drying out. And it looks like some of these cells may be producing these compounds with humectant like properties to actually pull water vapor in to help resuscitate themselves or at least to stave off the effects of desiccation. So this was also quite a surprise for this project.
Reminds me a lot about cryoprotectants, that is to say, compounds that microbes make to survive low temperature or freezing.
Yes, I think there's probably a lot of um, parallels with that. These compounds may also be osmolytes, and
Mm.
I'm speculating here that one of the functions of osmolytes. So typically, we think of osmolytes as a way to balance the water potential inside and outside of the cell in salty environments. So marine bacteria, for example, produce or um, transport osmolytes from their environment to cope with the fact that they're in a salty environment. In desiccated cells, there's a lot of literature and interest in the idea that um, osmolytes are produced to, again, help resist the difference in water potential on the inside versus the outside of the cell. But maybe they have another role. Maybe they're involved in somehow pulling water towards the cell from the atmosphere, water vapor towards the cell. And obviously, our data is not going to be able to resolve this question, but it's something that we're very interested in, this ability of microbes to pull water vapor in. There's not anything I can find, or very little that I can find, um, that discusses this in different bacteria. So when we were analyzing our RNA data, we made this additional interesting observation that this waking up by water vapor seems to coincide with different transcriptome dynamics as well. So, at each of these four time points, we collected cells, extracted RNA, and sequenced them. When we plot those transcriptome profiles on a PCA plot, we observed this interesting phenomenon where at on day two, again, this is where the treatment and control are at 100% relative humidity. All the cells tend to have a distinct profile. And on this slide that I have here, they're all kind of clustered in the upper left corner. And upon desiccation, there's a shift in transcriptional dynamics. We expected this. So from day two to day eight, there's a big shift down to the lower left of this PCA plot. What we didn't expect is that between day 8 and day 14, so within about a week, there was zero change in the composition of that transcriptome. And there was a paper published by Jen Martini's group late last year that showed a similar finding in a different microbe. It's almost as if these mRNA profiles in the desiccated state are completely stable. So
Hmm.
the way I think about this, like I don't know, maybe some of you have seen Star Wars, is when they freeze Han Solo in carbonite, right? And he's locked there in carbonite. It's almost as if that's what's happening during desiccation to these cells. And those mRNA profiles are frozen in, not in carbonite, but inside of the cell. And then
Well,
when we
that's so in that
yeah, go ahead.
I was going to say that's so interesting because RNA is so notoriously labile. It tends to fall apart so easily, and its turnover is so rapid normally.
Yeah. So in actively dividing cells, RNA is turned over within minutes. In all sorts of organisms. This has been shown in E. coli, it's been shown in Prochlorococcus, it's been shown in other types of microbes as well. Um, and we use it as a marker of that, right? When we go hunting for active microbes, we look for RNA. We say, oh, if we see an RNA molecule, that means that microbe is active. Um, there's other techniques that use ribosomal RNA to ribosomal DNA ratios as a marker of activity as well. Where if there's a higher ratio where there's more mRNA, sorry, more rRNA relative to rDNA, that microbe is more active. In my opinion, this kind of shoots a little bit of hole in that. We can get RNA from cells that have been dried out for 14 days, and their mRNA profiles are pretty much stable in the desiccated state. We have a little bit more detailed work to do on this that won't be part of this publication. We're working on that now. It looks like this is not unique to this strain, and this might be a general feature of um, desiccation tolerant, at least, soil microbes. When we rehydrate these cells, this mRNA profile then shifts again to a new state. So the cells wake up by rehydration and they start transcribing different genes. And we've extracted sort of these temporal correlations and identified several different modules of gene expression where certain transcripts are abundant in the desiccated state, but low in the hydrated state, whether it's rehydrated or um, the initial state. Other transcripts are low in the desiccated state and higher in the active cells, and some of them have different patterns as well. And we're kind of digging into that data and trying to figure out what these modules mean physiologically. I think there will
Hmm.
be some limits to that a little bit, but that's kind of the direction that we're moving in this paper.
So this is really interesting. Everyone would have ideas of what would happen if you would dry something down and how quickly it goes away. And you went ahead and designed experiments to show what really takes place to regularize or better control for the process of dehydration and rehydration. You found this very unusual okay, maybe everyone knows about it. This happens to me all the time, by the way. This idea of RNA profile staying stable in these dry cells is fascinating to me because I've never, it makes me wonder if in the process of completely drying out, there's some kind of structural aspect that's helping protect the RNA. You don't know. I mean,
We have
certainly
no idea.
when you dry down tardigrades, one of my favorite creatures, one of the few eukaryotes that I'm fond of, it's really interesting because it does the weird chaotic thing when its proteins get like settled in. So I don't know if it's related at all, but we tend to be real funny about what we consider to be difficult or extreme environments. So this is really fascinating.
Yeah, yeah, we don't know what happens to the RNA. So one of the big things new lab personnel here shortly will begin to look at. Um, we're curious if RNA in dry cells actually gets translated after it's rehydrated, or if those transcripts are broken down into ribonucleosides, and those ribonucleosides are then used to um, support new synthesis. We see evidence that RNA is broken down in dry cells. And I can't remember if this is on one of the slides, but we see all four ribonucleosides. So a ribonucleoside is a ribonucleotide without the phosphorus group on it. Um, we see them enriched in the two desiccated time points, but absent in the hydrated time points. And we think that this has to do with the um, structural changes that happen between the hydrated and desiccated state, and then the desiccated
Hmm.
to hydrated state. But our experiments weren't set up to really pinpoint that. So that's something that we're interested in figuring out as time goes on with this project. Yeah, it's
I just
really
remember
surprising.
I just remember when we were thinking about trying to study tardigrades a little bit in my little laboratory with undergrads, one of the problems that we had is how do you dry them down effectively? Well, you just told me, right?
Yeah, yeah,
And that makes a lot more sense based on just putting it in a vacuum chamber, right?
yeah, it,
So that's very interesting stuff.
yeah,
You know, we talked about this business of what happens to the RNA profile that you see within the desiccated cell really is interesting to me. Now, you did say that there's evidence of the RNA being broken down, right? So that's something that's really hard to figure out if there's stuff being directly translated. How about the other macromolecular components of the cell, like proteins? This takes us back to what I said about tardigrades with these chaotic proteins during desiccation.
yeah. So in our transcriptome data in the desiccated cells, intrinsically disordered protein, a intrinsically disordered protein transcript is the most abundant transcript in the cell. So we think that it may have a similar function that you would find in tardigrades. We haven't looked specifically at what's going on with protein degradation, but there are some studies that have looked at protein oxidation, for example, during the
Mhm.
desiccated state, and that's a major consideration. As soon as those cells rehydrate, there are chaperones waiting, there are proteases waiting to take care of those damaged proteins. Um, and in some of our data, transcriptional data, we see evidence that some of those chaperones might also be important in this process.
So, Paul, we talked a little bit about desiccation resistance, and I was curious, is it possible to observe changes in cellular morphology? I mean, I mentioned the ultra microbacteria in the ocean that shrink when they're being starved. Is there something similar going on here?
Yeah, so there's a study from maybe the early to mid 2000s that looked at a actinobacteria and desiccation and looked at it under a microscope. And um, actinobacteria are gram positive. They have a thick cell wall, and for the most part, they don't look that different. They look a little bit
Hmm.
shrunken, maybe a little bit like a raisin. Is how I remember the figure looking at. So
So they
sometimes,
get
yes,
wrinkly.
they get a little wrinkly, as you might expect.
Yeah.
I'm not sure about gram negatives. My suspicion is that they do shrink to a certain degree, but there are compounds that some of these cells produce and can sort of help stabilize the membrane. Things like trehalose, for example, is a big one.
Hmm. That's a really interesting question. I know that that's something that's interested our producer for some time is the texture of the surface of different things that we look at under a microscope or know about. And I know that using atomic force microscopy, you can get some idea about quote the texture. It's kind of surreal to think about. And as you mentioned, if these things are getting all, forgive me, raisiny, which I would
Mhm,
associate with being like in the bath too long, but
mhm.
I see the
Yeah,
principle. Maybe there would be a change in texture, which is kind of an interesting idea too.
yeah, yeah, for sure.
A lot of buzz in everyday life about this series of novels and the television series, I believe on Netflix, I might have that wrong, for the three body problem, where you meet these extraterrestrials that deal with stressful conditions through desiccation, and then they get like tossed in water to revive. This is very different than your situation. I would argue, in fact, that like slow rehydration would be the way to go. Speaking for the extraterrestrials.
Yes, for sure. It,
And something that I don't know if you can answer this, but most microbes don't exist by themselves. They're part of a community. And it would be really interesting to learn what a community does during this desiccation and rehydration, or whether this is just specific to the one. And I understand if you have to use one particular organism as a model system. Do you have any thoughts about community dynamics?
yeah, the complexity of that is at least an order of magnitude, probably several orders of magnitude more complex than what we're studying. There have been a
Yes.
number of studies in soil showing that drying and rewetting has a significant effect on microbial community structure by a
Mm.
number of different groups in California and Colorado, California. Um, so the community definitely responds to this. What we're learning through some of our other research is that individual related microbes have vastly different desiccation tolerances. So, in Bradyrhizobium, for example, we have some Bradyrhizobium that cannot tolerate desiccation, and others that are as desiccation tolerant as some gram positive microbes. So, even within related lineages, there's a tremendous amount of variability in desiccation tolerance. We don't know why that is. We suspect it's because they have different gene suites, or they're using those gene
Sure.
suites in different ways. Um, so there's multiple layers to this. There's sort of the per phylotype desiccation response, and then there's the per microbiome desiccation response. And then that's all layered, at least in soil, within the complexity and three-dimensional dynamics of the soil environment. Very structured. Water runs through it in different ways, which has different effects on those microbes. So all of these things are interacting with one another to make that question a little bit complicated to answer.
Also wanted to ask one follow-up because I did my graduate work on some of the fast-growing rhizobia. I was working with Cynorhizobium mellilodi, the Bradyrhizobia japonicum that nodulates, for example, soybeans. Are these Bradyrhizobia, do they contain nitrogen fixation genes and symbiotic genes?
So we use Bradyrhizobium diazoepicans USDA 110 as our sort of nitrogen fixing model. Those are not
Okay.
very desiccation tolerant at all. And we obtained some knockout mutants of Trehalose biosynthetic machinery, which is known to have protective effects during desiccation. And those aren't actually that desiccation tolerant at all, which you might expect. But some of the isolates that we have are very desiccation tolerant. And interestingly, they didn't come from our desert sites. They came from some of our more hydrated sites, I will say, at the top of the mountains just outside of Tucson. And they're as desiccation tolerant as the Arthobacter strains that we're working with as gram positive
Crazy.
microbes. So we're very interested in what's driving that, whether it's biofilm production or some other physiological response, but don't have any answers on that just yet.
I really think this is fabulous work and must be
Thank you.
very satisfying to have this like very interactive, very interesting crew working with you on it.
I love my crew.
Tell me about your crew.
Yeah, so we've got two PhD students now in the lab. Um, one of them is interested in culturing archaea and anaerobic archaea, really all archaea, anaerobic, and we have a different culture of ammonia oxidizing archaea that we've been struggling with from our desert soils. But um, uh, she's interested in that, Caitlin. And Adriana is an astrobiologist who's really interested in a lot of the questions we just discussed. She's very interested in how these cells are um, waking up from this desiccated state by using water vapor. And she's got some new data that indicates that, um, I explained how we dry the cells out and then we dunk them in a buffer to quantify how many viable cells there are. Well, she can show that 0 cells come back after 2 or 3 weeks of desiccation if you dunk them in the buffer. So it would look like they're all dead. But if you take that filter after 2 or 3 weeks and rehydrate it with water vapor, they all come back. So there's something to this length, duration of time that they're dried out for, and the way you wake them up that is important. And we're not quite exactly sure what it is, and we're not quite sure exactly how to study the water vapor effects. We have some ideas that we're pursuing right now, but there's something about this gentle rehydration that is very important for these cells. So she's working on that.
Do you know the quote from Leslie Orgel that Francis Crick made a big deal about? He said
Mm-mm.
that evolution is smarter than we are? Well,
Uh-huh.
actually, he said evolution is smarter than you are, which Leslie Orgel could say to me. That would be okay. Um, but I took that and I thought, and this is a really good example of what you're talking about. Microbes are smarter than we are. I don't mean that they think, but we tend to put human ideas onto them. So absolutely, very smart people. When I talked about the Viking landings, they thought it would be fine to just dump some soil into broth, or they said it's humidified, but again, they dumped some broth onto a soil sample. And what you just talked about is a situation where if you did similar things to that, you wouldn't recover anything alive.
Yes.
And no one would have guessed that, except you kind of thought, what really takes place in soil?
Yeah.
It's not dumping stuff in a tube.
Right.
And then you had to design the way to do that. So this is a good example where you bring lots of different people together with clearly have many interests in your laboratory group, and they all come together. And this is something we've talked about in the podcast a lot, that sometimes things can get too narrow. And I keep insisting that when you have lots of different people with different backgrounds, you get unusual and interesting ideas.
Yes. And one of our lab members, Dr. Una Snow West, did her doctoral degree, her double doctoral degree with Lynn Margulis. And we're fortunate to have her working with us on these projects and her ideas. We've got a great crew of undergraduates that we've just been really fortunate to hang on to for 3 years now. They're leaving it and graduating, not leaving, graduating in May. And we're all subtly anxious about that when they leave. What are we going to do? But I'm sure we'll find some new great undergrads. We have 2 new postdocs starting in the next month that we're really excited to see where they take our different projects in the future as well. So very excited about our lab group.
I think that you've really shown how important it is to have a kind of a diverse, interesting group of people working together with lots of different ideas. And again, something else we've talked about over and over again on the podcast is there's lots and lots of different paths to becoming a microbial scientist.
Yeah.
And you're a really good example of that. Do you have any fun stories about living in Tucson?
Oh, fun stories. I mean, it gets really hot here in the summer, and all of the wildlife comes out. We've seen tarantulas on our sampling trips and rattlesnakes. And if you're not careful, like me, when I'm sampling, you'll turn around into a cactus, and the cacti in the yards of all the houses have spines that are about that long. But once you get out into um, the actual desert, they're much longer. And I got a inner leg full of these 2-inch long splines that went through my pants. So I had to drop trowel in the field and get them out right there because they were pretty painful. Um, yeah, it's an interesting place to live. The winters are absolutely amazing here. It's beautiful, cool. You can do a lot of outdoor stuff. So we love the winters and the fall and the spring. Unfortunately, the next couple days it's supposed to be in the triple digits, so it's still a little warm. Um, but yeah, it's a great place to live.
Yeah, I've only been to Tucson a couple of times, and it was always a party to kind of critters that you would see coming across the floor of the house I was staying at. But you find different things wherever you go.
Yeah.
Do you? And we already covered what sold you on microbiology, and for you, it was kind of like digging in the dirt. By the way, that's an old Peter Gabriel song. So there you go. You're
There
all
you
set
go.
with that as a theme song.
Yep.
So the other thing that I was going to say is that it's been wonderful having you on the podcast. I've learned a lot, and this is one of the reasons that I enjoy doing the podcast. I often get ideas about some of the things that I do just from chatting with folks like yourself, and I want to thank you for that. And I want to wish you and your coworkers and family, whoever you're close to, the very, very best, and thank you for your time again.
And I want to thank you for having me on and for being such an amazing advocate for our discipline of microbiology. I have enjoyed watching many of your videos and following you over the years, so it's a pleasure to meet you and be a part of this show.
Well, I hope we get to meet in person one day soon.
Yes, thank you.
Thanks for your time.
Thanks.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with awe-inspiring links, as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Paul Carini is in the Department of Environmental Science at the University of Arizona in Tucson, Arizona. Many thanks to David Renata for superb editing and Rupert Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.